Dr. Konenika Ray is here, who is the co-director of this IBRC consortium and the co-founder. Then it is Open Source Pharma Foundation, and Professor Jaykumar Menon is the co-founder of this organization. And this is an association with Indian Phage Society called SBRT. And the office bearers of this society are Dr. Vinod from Karnataka, he's the president. Dr. Amita Gupta, she's the vice president from Delhi University. And Dr. Ramesh is from Vellore Institute of Technology, he's the secretary. So I thank all of you for co-organizing this event. And Dr. Schooley, we have been we really, really appreciate your time. It's early morning for you. And thank you for accepting our invitation. So may I please introduce you, though you are very, very well known and I'm very, but then this talk is mainly about phages. So just give me a minute. Let me introduce Dr.
So thanks, sir. I'm sorry. We're just having a little bit of trouble with the audio. Go ahead. I'm sorry.
Oh, you can't hear me well?
Now I can hear you better. Thank you.
Dr. Schooley is a professor in the Division of Infectious Diseases within the Department of Medicine at the University of California, San Diego School of Medicine. He is an interim faculty director, global education senior director, international initiative professor of medicine, Division of Infectious Diseases and Global Public Health co-director, and the co-director of Center for Innovative Phage Application and Therapeutics. This Center for Innovative Phage Application and Therapeutics, called as IPATH, was founded after the famous case of Professor Tom Patterson, who was treated by Dr. Robert Schooley in San Diego. And we all know this case which has been a very high-profile case in 2016. And Professor Stephanie Stradley has promoted this case so much that the whole world kind of took notice of this case. And now I think phage therapy is getting the traction it deserves. So Professor Robert Schooley's interests are in diagnosis, pathogenesis, and therapy of viral infections and in overall global health. And he was the first physician in 2016 from US to treat a patient, which I mentioned just now, Tom Patterson, who was given intravenous bacteriophage therapy in a systemic bacterial infection. And this case was then drew everybody's attention, it drew global attention. And from there onwards, we had this book called Perfect Predator and the IPATH. And I remember when we were watching this case study, then we got to know that Dr. Schooley dug up some Russian literature to know that how to deliver phages to a patient, because it was never done before in the US. That was very brave of you and very far-sighted. And because of that, a patient could get back to life. Thank you so much, Dr. Schooley, once again for joining in. And we welcome you to share your experience with us, please.
Thanks so much for the hospitality today, and good morning from sunny California. I'm very much looking forward to a discussion after the talk. Do I have the right screen up, or are you watching the, let me just get the appropriate screen up here.
I think your screen looks fine.
Problem is hold on one second. I'm going to go off. There we go. Okay. Okay. Do we have the right screen, or shall I switch screens?
You go to slide share, perhaps the screen would be more useful.
Okay, let me swap screens here. Let's see. Are you still seeing the, are you seeing the right screen?
I think it's fine. You can go ahead.
Let me see if I can switch screens. I'm sorry if we're,
Go to swap displays.
That's what I'm looking for.
Upper left. Yeah, there you go. Yeah, you got your cursor almost right on it. Swap displays, right? Just to the left of use slideshow.
Just to the left of slideshow.
Use slideshow. Yeah, your cursor is right, almost right there. Just move to the left a little bit to swap displays.
Yeah,
There you go.
Okay, thanks very much. I appreciate the expert help. All right, I'm going to try to spend the next 30 or 40 minutes talking about moving from where we've been over the last several years, which is making rapid advances in providing phage therapy to people around the world, to getting where we need to be, to having them be generally available in the context of mainstream medicine. My disclosures are here. And I'll start with something this group well knows. We're talking about a field that's now 100 years old, that has been kept alive by people who have been active in the Eastern Hemisphere, but is just coming back to being widely appreciated in the U.S. and in Western Europe. And why is that? Well, it's because we're finally, I think, recognizing that MDR and XDR infections are becoming more and more of a problem on a global basis. Traditional small molecule development has been slow and is not keeping up with the global resistome. And there have been major advances in biotechnology, phage discovery, phage engineering, and preparation of phages for clinical use that's made access to phages clinically much more feasible than it was 8 to 10 years ago. As has already been mentioned, the Tom Patterson case is something that got me interested in this. It wasn't something that I was anticipating getting involved in when Stephanie Strathty, my colleague at the University of Colorado, San Diego, called me about her husband, Tom, who had become ill while traveling in Egypt with what became a disseminated Acinetobacter infection. Without telling the shaggy dog story, to make a long story short, Stephanie ran across a paper about Acinetobacter phages from the Eliava group and asked if it would be feasible to treat her husband with these phages. And as described in her book, The Perfect Predator, that's indeed what we did. And after a number of months, he returned to health. And since that time, there have been a gradual increase in the number of phage case reports in the medical literature. Many cases obviously go unreported. But this is in some ways a turning point, not the case itself, but I think the time in which the case occurred that enabled phage therapy to be more widely used. These cases that are being reported include treatment of implanted infected devices, joints, cardiac devices, and other infected implanted devices that get coated with biofilms, pneumonia, osteomyelitis, multiple miscellaneous sites. And these have been really instructive to us in terms of beginning to give us an idea about what types of infections might benefit from phage therapy. But they're biased by the fact that they're anecdotes. You could argue that the anecdotes that get reported are the ones that are most likely to be successful, and therefore the ones that one would want to follow up with on clinical trials. These have occurred around the world. If you think about kind of where things were a decade ago, there was a lot of work going on in Europe, in the Middle East, and elsewhere. But this, in the last 10 years or so, more and more centers have begun to pop up in the U.S. and in the Western Hemisphere as well, to complement the work that's been going on elsewhere. And I think this is going to continue over the course of the next several years. The real question, though, is how do we get from where we are now, which is clinical anecdotes, to widespread clinical applications? And the take-home message, really, from my perspective, is we have to think about these as living antibiotics and to investigate phages clinically just as we would if someone handed us a vial that had a small molecule in it and said, I want to learn how to use this in patients. Clinical trials need to be designed using the same principles, using the same metrics, and using the same sequence that we would use to develop an antibiotic. for two reasons. One is this approach has worked in the past in terms of giving us insights about how to treat infections. And secondly, we're going to be using phages in the context of other antimicrobial agents, particularly in the context of antibiotics. And we need to understand how phages work using the same metrics we would use to assess an antibiotic. I'm going to try to address a handful of issues today. These are by no means all the issues, but there's some of them that we can talk about. And it just illustrates the fact that we've learned a lot, but there's a lot more that we need to know. How do we know what phages to use? How do we combine phages with themselves, with each other and antibiotics? How should we administer them? Can we improve phages by molecular engineering? How does the adaptive immune response compromise phage activity if it does? And how should phage therapy be regulated? And let's start with how to decide what phage to use. If you think about antimicrobial testing for antibiotics, we've spent about 40 to 50 years going from what we now view as rudimentary and somewhat often misleading approaches to ones that are much more refined, but it's taken a long time to be able to develop the laboratory correlates of clinical utility. Phage susceptibility testing is just entering this phase of clinical validation. It's going to take some time for us to be able to understand how best to interpret phage susceptibility testing in vitro and apply it to clinical applications. As this audience well knows, the two most basic approaches to phage therapy are traditional plaque assays and ones in which phages are grown on their bacterial host and liquid medium, and growth is assessed using one of several different types of markers. Both of these seem simple, but in fact, they really do require quite a bit of interpretation, even to get the in vitro understanding of what is being looked at. I would refer the audience really to, I think, what a very nicely written review of this that just appeared in a supplement in Clinical Infectious Diseases by Ron Nearpaz, Ronan Hazan, and their colleagues that talks about some of the challenges and opportunities in understanding how to interpret these results in the context of phage susceptibility testing. In this article, they talk very much and with great insight into the complexity of trying to understand how to assess growth curves. It's pretty easy if one sees in the left panel, for example, phages that are able to completely suppress growth of the bacterial host for a prolonged period of time. That's a very easy read, but it becomes a little bit more complicated when one starts thinking about bacteria that are suppressed for a while and then begin to regrow and have other patterns of growth. Does one try to look at the suppression of bacterial growth at a specific point in time? Does one look at the ultimate outcome at the end of the course of therapy in vitro? Or does one try to assess the area under the curve? These are all very legitimate ways to assess suppression of growth in vitro, but we don't yet know how to extrapolate these to what it might mean in a clinical setting. This is very well described in this manuscript. I will not go through this table, but I think it's really a very poignant way to look at some of these complex issues. Now, when one starts, once one gets through phage susceptibility testing in one laboratory, you start to have to thinking about reproducibility and extrapolatability to other laboratories. This is just beginning to be done. This has been done for a very long time in antimicrobial testing with exchanges of strains, exchanges of reagents, and refinement of technology. And we're just beginning to get there with phage susceptibility testing. Not much has been done, but in general, one could say that if the same assay is repeated in the same laboratory, the results are generally pretty overlapping. They're not totally synonymous, but they're pretty good in terms of reproducibility. If you try to do the same assay in different laboratories, as in antimicrobial testing, still pretty good overlap, but not quite as good. When you start using different assays, for example, trying to assess phage susceptibility by plaque assays compared to liquid growth assays, what you'll find is that most phages, if they are susceptible in one assay, will be susceptible in others, but not insignificant number of phages will be suppressed. Bacteria will be suppressed in a plaque assay, but not a bacterial liquid phase growth assay, and vice versa. And we don't yet know the implications for that. And then when one starts trying to compare across laboratories, different assays, it becomes even more complicated in terms of being able to understand what these results might mean clinically. These are problems both in terms of thinking about phage selection in individual cases as we speak, and also in clinical trials as we move forward. And this is just trying to assay lytic activity of phages. We also know that phages are used to try to disrupt biofilms on implanted devices. And with phage engineering and other approaches, people are making attempts to improve the ability of phages to disrupt biofilms. And we don't yet know how to do clinically meaningful assays of biofilm disruption with phage in vitro. Now, the holy grail, of course, would be try to understand how one might be able to use molecular testing to predict phage activity. Are we there yet? Not yet. Will we get there? Eventually we might, but it's going to be complicated for a number of reasons, not the least of which is that phages and bacteria have been living together for 300 million years and developing approaches to, on the part of the bacteria, to avoid phage predation. Phages, on the other hand, have been trying to get around these blocks to phage predation. And so any given bacteria may use one of many different mechanisms to avoid being lysed or attacked by phages. And if one is thinking about a molecular assay that's going to try to understand whether or not any or all of these mechanisms are present, the informatics get quite complicated. It's quite possible that over time, though, we'll begin to crack this with some of the simpler organisms, but it's not going to be easy to get around and understand on a molecular basis how to be able to read all of these mechanisms in any given organism over a short period of time. Now, there are ways that one might think about using molecular approaches to choose phages, and that might be in using phages in public health applications for outbreaks. We recently had an experience collaborating with the Outbreak Investigations Branch of the US CDC, looking at Pseudomonas contamination of eye drops that have been made without preservatives. This has been an ongoing problem in which these eye drops have been contaminated, 2 different lots initially, and now up to 26 different manufacturers. There have been, even by last spring, over 80 cases in 18 US states, with quite a bit of morbidity in terms of vision loss and occasional deaths. These Pseudomonas isolates are highly drug resistant and are highly invasive and have been problematic in multiple cases. Now, the bad news about these is these are very highly resistant to antibiotics. This is an antibiogram of 3 of those isolates that David Pride obtained from the CDC, showing you that they're difficult to treat with traditional antibiotics. But the good news is that when one starts screening for phage activity, as he's done here with some jumbo phages, this is a depiction of a number of Pseudomonas strains across the top. These 3 are 3 Pseudomonas strains from this outbreak investigation. These are a number of phages with activity against pseudomonads from David's laboratory. And what you can see is that there are phages that have activity against these outbreak strains, which are close to monogenic. And so if one thinks about outbreak investigations, this might be a situation where one could develop a small library of phages, and when an outbreak is discovered, just doing a simple molecular test to see if a case is associated with that outbreak might be a way to move quickly to phage intervention without having to go to phenotypic testing. It gets even more complicated when one starts thinking about trying to combine phages with each other, moving from just phage susceptibility testing with one phage at a time, and with trying to combine phage with antibiotics. When we were initially trying to think about treating Tom Patterson, we were anxious to have more than one phage in the mix because we were concerned about the emergence of resistance to the phages given the organism load that Tom was dealing with. And the way the investigators at the Biomedical Defense Research Directorate, the Navy, went about this was to take their phage library that were active against Acinetobacter and to do a checkerboard titration against multiple Acinetobacter strains and pick phages that were able to suppress outgrowth of multiple different ones of these Acinetobacter for at least 12 hours. And they found about a half dozen unique phages that had activity against different Acinetobacter strains, and assumed that these would be phages that would be different enough to put into a cocktail and be treating the patient with a phage cocktail that would be resilient to prevent emergence of resistant phage bacteria, at least over a reasonable period of time. In vitro, this looked quite good. You can see here that the individual phages or the phages in combination suppressed outgrowth of the Acinetobacter for 20 hours in this in vitro assay. Now, when the phages were used in the clinic, up in the upper right-hand panel is the graphic I just showed you. But within 6 days, and certainly about 10 days, you can see that the bacteria were growing through each one of these phages and had become highly resistant to each of the phages. Now, before we used these phages, we did not have the sequence of any of them. This was back in the dark ages of having phages that had not yet been molecularly characterized. There was even difficulty initially sequencing these phages because they had methylated their DNA, and the DNA kits were not doing a good job at the time of being able to do the sequencing. So we were working with unsequenced phages, but when they were sequenced, what we found out is that they were very similar to each other. In fact, they differed just in a minimal degree, and we were probably just looking at minimal differences in the binding epitopes of these nearly identical phages. Biswas and Theron Hamilton were able to go back and then take that isolate of Acinetobacter baumannii that had been obtained 10 days into Tom's treatment, and then go back and look for phages that were active against it, and found a phage combination that could suppress the new isolate, validating the fact that one can come back with separate rounds of phages. And when we looked at that, it turned out to be molecularly quite a different phage. So as we've learned more about this, it's going to be, I think, quite clear that we're going to have to be a little more creative about thinking about how cocktails get put together. Now, before I get off of phage susceptibility testing and selection for phage resistance, I'd just like to make one side comment, and that is that it may not be all bad to have phage that have selected for resistant organisms in a clinical case. In Tom's case, what you can see is that his initial isolate was very juicily covered by capsule, and by the time 10 days later, this capsule had been lost as the phages selected for mutants that lacked the capsule, and by going back and doing molecular characterization of the isolate that evolved from the initial isolate, our colleagues at Texas A&M were able to demonstrate that the major differences were in parts of the genome that promoted growth of the capsule, so that selection of phage-resistant bacteria may well lead or be associated with less virulent, less invasive bacterial pathogens that can be better handled by the host immune response. In this case, these isolates were less invasive in a silkworm invasion assay, and presumably were better handled by the patient's polymorphonuclear cells. Now, combination testing again, getting back to the paper from Ron Nearpass and Ronan and their colleagues, is also something that requires, I think, quite a bit of careful thought. When some of these growth curves are really fairly intuitive. When one looks at phages that are neutral, you can see that this is the control result. This is agent 1 can be a phage or an antibiotic, and agent 2, and you can see that these tend to be neutral, in this case, synergistic or additive. And then there are ways to look at things so that when the sum is worth less than the parts, you start getting into antagonism. And we don't yet know how to interpret these results and use them in putting together phage combinations for in vivo use. But these are things we're going to have to learn in clinical trials. The other challenge is going to be that many of these assays are done under very carefully controlled conditions in which ratios of phages and ratios of phage and antibiotics are carefully controlled, growth media are controlled, bacterial growth conditions are controlled. And in vivo, things are really often quite different in terms of ratios of phages to each other, ratios of phages to antibiotic concentrations, changes over time. And one can expect this to be much less clean when one starts trying to go directly from a nice laboratory assay to what goes on in the clinic. It's going to be very important to try to connect these dots if we're going to be able to use these assays intelligently to concoct combination regimens. Now, there have been some very creative ways of putting antibiotics together with phages, and Paul Turner and Ben Chan, this is a very classic case done about the same time as Tom Patterson, in which they had a patient who had an infected aortic Dacron graft that was not operable because of its anatomic location and the state of the patient, and was not sterilizable with antibiotics because of the biofilm. They had the insight to select a phage for activity that targeted the efflux system of the Pseudomonas that was infecting this patient, and prevented the Pseudomonas from pumping antibiotics out of itself as part of its defense mechanism against antibiotics, and resensitized the organism to the antibiotic that had previously been unsuccessful, and were able to cure the patient. This approach, I think, is one that was brilliant at the time, remains brilliant, and something we need to learn more about to be able to generalize it and perhaps find ways to make this a more broadly used approach with different antibiotics and different phages. And then finally, to go to Wayne Gretzky, a famous American hockey player, whose one of his famous quotes was, "A good hockey player plays where the puck is. A great hockey player plays where the puck is going to be." Predicting where phages will push an organism and thinking about how to combine phages so you'll have one to be there when it gets there is something that the Wayne Gretzky of phage therapy, Anthony Moresso, and others have been thinking about. Can one use what we know about phage evolution to concoct phage cocktails that will move in the direction of cutting off escape mechanisms that particular organisms have to phage selection? We've done some of this in antiretroviral therapy with agents that are active against the reverse transcriptase. Phage and bacteria are much more complicated, but the principle can certainly be the same. How do we know how to administer phages? What's the route, dose, duration? Well, for a long time, we were kind of stuck with having to use phages topically or giving them orally because of endotoxin and other contaminants that were present in phage preps. We've now been kind of liberated and can give phages by those routes as well as by intravenous routes, intraoperative routes, nebulized routes, basically multiple different approaches to phage administration because of advances in phage purity. And we don't yet know the best way to use them. And in Tom Patterson's case, we decided that we needed to use them systemically because he was so his infection was so widespread. We were isolating organisms from his sputum, from his intra-abdominal abscess cavities, from his urine. And there wasn't really any specific place we were able to give phages that would cover his entire anatomy, and decided that we would try to give them intravenously with the hope that the bloodstream would disseminate them, they would find their bacterial niche, grow there, and be able to be therapeutically active. How much did we give? In that case, we actually had to back titrate from how much we could give based on the residual endotoxin in the phage prep. The decision to use 10 to the 9th plaque forming units wasn't because we had any insight mathematically that that was the right dose. It was just the highest dose we could give at the time, given phage purity limitations. This dose has been used quite a bit in many patients since that time, but we still don't know whether or not this is the optimal dose in any specific infection, and certainly not for multiple different types of infection. Much more work is needed here. In Tom's case, we gave the initial phages we obtained from Texas A&M into his abdominal abscess cavities through catheters. He systemically stabilized somewhat, but certainly didn't improve markedly until we moved a couple of days later to intravenous phage therapy, which is when he really turned the corner. Now, clinical trials are just beginning to look at this in an organized way. Pranita Tama and I are doing a study with support of the NIH's antibiotic resistance leadership group, looking at several different doses of phages in a group of patients with stable cystic fibrosis who are shedding Pseudomonas from their sputum. This is, at this point in its evolution, just a single dose study comparing 3 different doses of phages and looking at shedding of Pseudomonas as its output in the sputum over time. I think we're going to need to go to multi-dose administration as we analyze stage 2A, which is where we are now in the next month or so, because I think that it's going to be necessary to give multiple doses to be able to reliably get phages to the sputum by this route. But this was the start of this multi-dose study to try to look at that. In a study that was presented at ID Week in Boston about 6 weeks ago, Biomics did a very nice study that looked at essentially the same approach, same patient population, but gave the phages through a nebulized approach over a 6-day period. They randomized patients to phage or no phage, and in a placebo-controlled way, and started the phages either in patients who were on continuous antibiotic suppression or in those who were in cycling antibiotics with cystic fibrosis as they went to a new cycle. They did 2 things in this study that I thought were really very important from the standpoint of understanding phage use. First of all, they looked at this phages in the sputum, and what they found is that phages could be found in the sputum throughout the period of therapy. And this is a very important part of PK/PD understanding that should be something that we try to do in every clinical trial that's done when the clinical infection site is accessible, so we understand what's going on at the site of infection. The Fericity study in burns was a study that failed, but it was a brilliant study from the standpoint of at least looking at many of these parameters that helped us understand how to do the next trial better. The second thing about this study was they were able to show that in most patients who received the active cocktail of anti-Pseudomonas phages, on day 15, there were 2 to 3 log drops of Pseudomonas in the sputum, 7 days after phage therapy had been stopped compared to the placebo. So this was very nice evidence of phage activity from the microbiological perspective. And they had early evidence of changes in FEV1 of the patients who received phages compared to those who didn't. So from the standpoint of a clinical trial, this served many purposes. It demonstrated phages at the site of infection, demonstrated impact on an important biomarker, and it demonstrated early evidence of improvement in the clinical disease state being treated. And these types of studies in which we began to be able to link microbiologic endpoints to clinical biomarkers to clinical outcomes are the ones that are going to help us, that will help us design intelligent phase 3 studies that get phages much more broadly into specific patient populations. Can phages be engineered to improve their activity? Can we give engineered phage? Well, giving engineered phage is actually something that is being done relatively frequently now. The first engineered phage to be given was one that Graham Hatfill and his colleagues organized with Helen Spencer. This was a young woman from the UK who had cystic fibrosis and an abscessus infection that recurred after her lung transplant and was not responsive to antimicrobial therapy. Graham had been running a program called C-Phages for a number of years with support from the National Science Foundation and the Howard Hughes Foundation, in which undergraduates in U.S. universities screened for phages against Mycobacterium schmegmatis as part of an early course in microbiology to understand evolution. They screened for these phages, they sequenced them, they named them, they sent them to Graham, and Graham had 15,000 of them in his freezer when he began to look at their activity against other non-tuberculous mycobacteria and found that some of them, in fact, had activity against M. abscessus. The problem with them is that, like most mycobacterial phages in nature, these phages were lysogenic and would not have been good phages to use therapeutically. What Graham had developed as well in his laboratory, however, was a technique called bacteriophage recombinering, in which he co-transfected sequences of DNA into permissive mycobacteria with phages that he wanted to engineer, selected for plaques that had shorter DNA in that segment, and was able to purify phages that had been engineered to lose their integrase and repressor genes, one or the other or both, and make these phages that had been previously lysogenic lytic. In this case, they were used clinically. The patient responded both in terms of her skin lesions and in terms of her pulmonary function tests. This got over the theoretical hump about at least using engineered phages in clinical medicine. Now, a lot has happened since that time, and there are many other ways to engineer phages. This is a figure that I won't go through in detail from a paper that Stephanie organized with several of us in Cell earlier this year, that has a discussion of some of the ways in which phages can be engineered. And a number of companies and individual laboratories are now engineering phages with fingers crossed, hopefully improve some of their functionality. For example, as I already said, making lysogenic phages lytic, improving host range, making phages more effective bacterial killers, finding ways in which phage might be able to disrupt biofilm in a more effective way. These sorts of phage engineering approaches are underway. Some of the ones, particularly the CRISPR-Cas phages, have found their way into phase 1 studies in people, and we're moving further into clinical trials. And as these occur, we're going to need to see if these improvements actually, these theoretical improvements actually make improvements in their clinical utility using the same sorts of metrics that were used in the biomics trial that I showed you just a few minutes ago. Moving along, what about the adaptive immune response? Well, immunologists for many years have used phages as a laboratory tool to understand the animal and human immune response to viruses. They can be injected harmlessly, and antibodies generally are produced. And we know in phage therapy that in some patients, antibodies to the phages have been observed. We don't yet know in great detail what impact this has on outcomes. We're beginning to have some idea about that in some clinical trial series. This is a series of patients that Graham Hatfill and Constance Benson published in Clinical Infectious Diseases last year that looked at 20 of the first patients with drug-resistant mycobacterial infections that were treated in this collaboration. And to cut to the chase, the success was seen in about half the patients who were treated. But when one looked for antiphage antibodies, 8 patients developed antiphage antibodies, and the evolution of these antibodies seemed to have a negative response on therapy in about 4 patients and didn't seem to have an impact on 4 others. We'll need to understand better how often this occurs, if there are ways to get around it, what patient populations are more likely to have this. Would this be more likely to be in a patient, for example, with bronchiectasis, with an intact immune response, and long-term non-tuberculous mycobacterial infection, or a patient with a lung transplant for cystic fibrosis or for some other indication? Are there ways to get around this in terms of engineering phages as we do monoclonal antibodies to make them less immunogenic? Can we use phages of different classes to get around this if phage antibodies evolve and one needs to get away from them? And then finally, how should phage therapy be regulated? This is something that gets discussed everywhere, and it's something that has evolved in different ways in different places. In general, in Europe, the magistral approach has been used. This has been something that Jean-Paul Pernet and others have pioneered, in which regulation is really more local than central. In the US, we're regulated by the US FDA, and we have a very straightforward regulatory pathway that allows us, if a patient is extremely ill, as Tom Patterson was, to essentially pick up the phone and request an emergency IND from the FDA. And in this case, you just described to the FDA the patient, you talk to them about what you know about the infection, the phage preparation you want to use, and you will be talking to someone like Cara Fiore, who is an FDA regulator, who understands phage therapy and can give you approval over the phone. If the infection is one that's a bit more chronic, for example, an infection that involves a prosthetic joint, and one has time to curate the phages in a more effective manner, then a regular IND is requested. You have to go to your local IRB, spend a lot more time talking to the FDA about phage purity and so forth, and then move on and consent and treat the patient. But this streamlined approach has not been something that has gotten in the way of therapy of patients who are in extremis. Now, we're moving on to clinical trials as we speak. They picked up their pace at about the same time case reports did, which is fantastic, because this is going to be the way out of where we are, of all these anecdotes to more widespread use. And as we go off and move from clinical trials to kind of the great beyond, I think it's going to be important to be very careful about how these clinical trials are done. Because a bad clinical trial can set the field back quite a bit. A good clinical trial may or may not be successful clinically, but if it's done in a way in which we learn something about why it works, if it works, and if it doesn't work, what the problems were, we're going to be much more able to move the field forward and be much more capable of doing the next trial in a better way. The way HIV therapeutics moved us from having patients with a fatal disease to one in which the patients live for almost as long as uninfected patients do with HIV was a trial matrix in which we iterated between things in the laboratory and things in the clinic, activities in both locations, and used translational tools to tie the two together in a series of iterative clinical trials in which New agents that had been discovered in the laboratory were put into clinical trials that included adaptive designs and others that allowed us to quickly assess biological activity using endpoints that let us look at, in the case of HIV RNA. In the case of phages, one can quickly look at the bacterial target with most anatomic sites of treatment, understanding PK-BT relationships and tying them to clinical endpoints, using mathematical modeling tools to understand quantitative relationships among these critical outputs, and then going back to the laboratory and tweaking things to have things improved for the next round, and always trying to use them in collaboration and coordination with best available other therapeutic alternatives, including antibiotics and other entities that develop in parallel. So a lot of ways that phages are going to be used, both in medicine and biology. I think we have a bright future ahead of phage therapeutics as they move into their second century of human interaction and their 500 millionth and first year of time on the planet. So I'll stop here, and thanks very much. Wish all of you a happy holiday season from my colleagues here at IPATH, and look forward to interacting with you going forward. I hope we have some time for questions here. I apologize for going quickly, but I realize that time is short. So I'll turn this back over to our moderators. Thank you.
Thank you so much, Dr. Schooley. That was very comprehensive. I think you covered almost everything. One thing, if you could speak a little on the microbiome targeting of phages, if that could be touched upon, because what I'm seeing these days in conferences, this is the most hot topic, that how can we specifically target the bugs in microbiome by phages and perhaps take care of some non-infectious diseases?
No, that's a great question. I didn't address that today. One of many topics I didn't address. But the elegant part about phages and targeting the microbiome is that phages are more like a laser than a hand grenade. People have tried to manipulate the microbiome with antibiotics in the past, and what this leads to is such disruptions of the microbiome that one ends up not being able to scientifically assess what is going on. There are a number of clinical entities, including inflammatory bowel disease, accelerated liver failure and alcoholic liver disease, in which individual bacterial species have been hypothesized to be the driving force, and phages are one way to look at that. People have thought about trying to curate the microbiome in the GI tract of people who are about to undergo chemotherapy for cancer to try to eliminate, for example, fluoroquinolone-resistant E. coli. And these approaches, I think, are very important. They may turn out to be clinically useful. It may turn out that we're just playing whack-a-mole and knocking down most of a given species and having the rest of them grow back up. But we have the tools to evaluate that. And as these trials are done, in addition to seeing if they have an impact on the clinical outcome and whether the underlying hypothesis is correct, it will be, I hope, a way to understand a lot more about quantitative relationships between phages and their prey in large populations in vivo. And so I see this as a field that should be investigated from the standpoint of people who want to understand pathogenesis, from the standpoint of clinicians who want to treat some of these difficult to treat, perhaps microbiome-driven diseases. And I see it really as a fantastic playground for mathematical modelers. So all for it.
Great. Thank you so much. So you can see a very good attendance for this hour of the day for us, so almost 100, and some of them have joined you on YouTube also. So I'll take a few questions for you now, that those which have been mentioned here. So first, okay, where has it gone? Just a minute. So one question, like you mentioned two case studies of mycobacterial infections, but they were non-mycobacterial infections. So one question is on the fate of phage therapy for tuberculosis. So would you have anything to say on that?
This is something that is of great interest, obviously. Some of the phages that Graham Hethel has are active against M. tuberculosis. And he is in the process of organizing some clinical studies to see whether or not this might be a useful therapeutic in the setting. There's been a lot of, I think, theological discussion about whether or not phages will access intracellular organisms and whether these studies should be done before animal studies demonstrate activity. And I would argue strongly against that because I think that the proof is in the pudding. It's easy to do early bactericidal activity assays with anti-tuberculous drugs. That's how we investigate all new drugs. And if phages are able to reduce mycobacterial load as a new anti-mycobacterial drug does over a 10-day period in vivo, you've got your answer about whether it accesses where the bacteria are in vivo. If you spend 7 years with mice, rats, dogs, cats, and primates doing these studies, you're still going to want to study this in humans. So I hope that these studies move ahead and don't get sidetracked by months and months of and years of animal model studies. I think they're very important to do.
Thanks. Thank you. So this one question is, phages and antibiotics sometimes work better when administered in an order. Did you try the order treatment approach while curing any infectious disease? This is from Rutuja. So I think in Tom's case also you found a delayed resistance to antibiotic, and which order did you follow, or was it given in synergy?
In his case, we were kind of stuck with, he was on about 3 antibiotics that had been shuffled for months in his case. And minocycline had plus minus activity here and there. After phages were added, the minocycline, they actually seemed to be more active against the minocycline than before, but I'm not convinced in his case we really did reverse susceptibility. I think in other cases, this may occur more often. Again, I don't think we know the answer to that question, and we need to study it in clinical trials to be able to begin to understand these sorts of relationships.
Dr. Shahid Jamil asks, can lytic phages be engineered for tropism, or is it easier to engineer a specific lysogenic phage to a lytic one? Which option is easier?
I think both can be done, and phages can also be genetically engineered the old-fashioned way from the standpoint of how we've engineered in agriculture for the last millennia. And that is by selecting for phages that have broader host range or a different host range by coevolution in vivo. So there are different ways to do this, either with directed molecular evolution with some of the tools I talked about, or by just co-growth in vivo. I don't know that one is easier than the other. I think that it may well depend on the organism and what you're trying to evolve and what you're trying to change. I think all approaches need to be investigated, both in the laboratory and then taken to the clinic to understand if they achieve what we had hoped.
This is from Tessa. Are there any organ function tests mandatory for phage therapy before administration or for topical use? Are there any organ function tests?
That's an easy answer. No. They're really one of the things that as long as the phages are produced in a way that endotoxin and other impurities are out of the way, they have been really remarkably non-toxic. In talking to colleagues at the FDA, kind of at different around different plants at meetings, they basically say we don't want to see a bunch of studies of safety. We want to see if they work. Yeah. So I think we're all convinced phages are safe. We're phage factories ourselves. So you can think about us as basically being ocean reefs that grow bacteria and phages in our GI tract for the convenience of microbial evolution. And we shouldn't worry about adding 10 to the 9th phages to the billions we carry around.
Yes, that's right. Okay, there are a few questions on lytic enzymes from phages, endolysins. So we hear a lot about phages, and Vincent Fischity says that once there's a single compassionate use of lysins, then perhaps they'll get the attention which phages have got in the recent past. So what do you have to say about the prospects of enterolicense?
I think license are a very legitimate area of research, and there are clinical trials ongoing with license, particularly in Staph aureus. License for gram-negative organisms a little more difficult to deliver parenterally. The upside is that they have a broad host range, and they can be given with little toxicity. The downside is they're like a drug, and you have to give them parenterally continuously, repetitively. They don't have the self-replicating benefits that you have from phages. But again, I think that clinical niches for them, either with antibiotics, with phages, or both, are things that we're going to see investigated over the next several years.
Right. Okay, this question is very often asked about the immune response. So you have touched upon it, but the question here is, what about innate and adaptive immune response to phages, and why multiple doses of phage preparation is required in clinical settings if phages are self-replicating in nature?
Both good questions. So the adaptive immune response does occur. Both cellular and humoral responses have been measured in vivo. As I mentioned in the talk, we don't yet know what impact they have on clinical outcome. It may depend on the anatomy of where the infection is. It may matter less, for example, if the infection is on the surface of a wound as opposed to on a heart valve, and it is going to take some time to kind of unravel that. Are phage immune responses more likely to be induced by humoral administration or parenteral administration or topical administration? Again, something we don't know. A trial that really needs to be done is a head-to-head trial of nebulized phages versus intravenous phages for example in patients with cystic fibrosis, to understand whether one works better than the other in different clinical settings, and whether or not one is more or less likely to get local or systemic immune responses depending on the site of infection. Now, why don't phages stick around when you infect a lawn? We don't know whether they do or don't yet. I think that some of these clinical trials that we're doing now will give us a better idea about what happens to them. Some infections, for example, like the one Tom Patterson had, really do involve someone with essentially a total body lawn of bacteria. But there are some other indications, for example, if you're treating a prosthetic joint infection where you might have a few colonies of staph here or there on different parts of the device in which you can't count on contiguous spread. So in somewhat of a backwards thinking, you could argue you have to treat more vigorously a smaller population of bacteria in the presence of a prosthetic joint infection or prosthetic treatment. But again, these are all things that we can talk about theologically. We have to study them in clinical trials and understand that. It's important to know whether they do stick around for an indefinite period of time, and if they do, how long that is, because that's how we decide about duration, about dose frequency. Again, most dosing has been done 2 to 3 times a day, sometimes 4 times a day in parenteral applications, but this has not been done because they've been compared to each other or compared to even daily use. So I think a lot more needs to be done to understand this.
Right. And Dr. Kaushal, we have made you can unmute yourself and ask a question. Dr. Kaushal has, again, though Dr. Schooley has talked about side effects, that there aren't many in terms of safety, but he wants to know, Dr. Kaushal, you want to speak? We have made you, you can unmute yourself. Okay, so then I'll move to the next question. This is from, yes, please. Yes, please.
Hello, Dr. Kaushal this side.
Yes, please.
So, ma'am, hello, sir. So, can be applicable in fungal infection also?
Can we treat fungal infections?
Yes, fast therapy can be.
Not at this point. That's not been something that has been focused upon.
So, this can be also used in TB case, tuberculosis?
For tuberculosis, to my knowledge, has not yet been treated. There are movements to Graham Hatfield's making movements to move in that direction in some early clinical trials, and I look forward to seeing what those results might be. But fungal phages are not things that have been worked with or are well known. So I think most of the virtually all the activity has been against bacterial and mycobacterial pathogens.
Okay. Thank you, sir.
Yes, sir.
And Dr. Swati Shom wants to know if any chances of phages recombining with other phages that already are there in the patient once you administer a dose or doses.
Yes, there's a possibility of that. There's also a possibility of, in the case of lysogenic phages for DNA exchanges. This is something that happens all the time. But that's something that is happening as I'm sitting here talking to you in my GI tract. And not something that we can avoid. So it's one of the things that we try to do in phages that are used for clinical use is to look at them for evidence of antimicrobial resistant sequences that they might be carrying with them, or sequences that are associated with toxins. But recombination is something that is part of microbiology.
Okay, a few more questions, Dr. Schooley. This is Dr. Gopalnath from India, and he also does phage therapy on diabetic foot ulcers and several such cases. His question is on septicemia. In case a patient has septicemia of different duration, then what should be the dose so that there is no sudden lysis which causes the toxin release or septic shock? So how do you go about dosing those patients? I think you've gone mute, Dr. Schooley, Dr. Sid Schooley.
No, can you hear me? I think it was my internet was unstable. That's been a question people have asked, but it's not been something that's been observed clinically. There was a very nice paper done by the DeBarbeau group and published in Clinical Infectious Diseases about 7 years ago, in which they in vitro looked at the rate of killing of two different bacterial species with phages or two different classes of antibiotics, and showed that with phages that killed as fast or faster than antibiotics, there was no difference in the rate of endotoxin release. So there's no evidence that phages are worse than antibiotics in terms of endotoxin release. And in people who are septic, they're already swimming in endotoxin. And endotoxin is tachyphylactic, so you're not going to be adding to a risk of anaphylaxis by using a phage in someone who already is septic.
And here, Dr. Ankush Gupta wants to know from BHU that what kind of molecular characterization is important for using phages for therapeutic purpose?
If one isn't dealing with an emergent situation, optimally the phage should be fully sequenced. It should be interrogated for the presence of genes that are associated with integration to understand its lifestyle. One should look at whether or not sequences associated with antibiotic resistance or toxins are present. And if one is looking at trying to use phages in combination, beginning to understand relationships of phages to each other would be something that would be good to do as well. But whole genome sequencing is really the best way to go.
Right. And I'll take a couple more questions. And after that, I mean, you can tell me when to stop because these questions your talk has evoked so much response that too many questions. Dr. Sanjay Biswas wants to know the effective delivery system for what in your opinion are the best delivery system for phages?
Well, intravenously, what we've used mainly here, Saima Aslam has been the person who's done most of the refinement, has essentially been to have patients self-administer phage at home through a PICC line using just a simple syringe over a short period of a minute or less. We don't worry so much about giving phages IV push. From the standpoint of, we generally give phages to a patient the first dose in an observed setting in which we could intervene if there were a problem, but we haven't seen a problem occur yet in the patients we treated. For aerosolized administration, there are a number of different nebulizers, some of which can destroy phage activity. So it's important to test the nebulizer that you're going to use with the phages you're going to use to make sure that these phages are not inactivated by the nebulizer. The phage administration that was used by Biomics had a specific nebulizer that seemed to be quite easily used by patients at home, in which the phage prep was just added just before the nebulization. So there are a number of ways to give them by an inhaled route. Phages have also been used topically on wounds, and in orthopedists like to slosh phages and antibiotics, for that matter, on implanted devices. I don't try to dissuade them, but I also don't try to encourage them, because one can't assume that you're going to envelop an entire device by splashing antibiotics on them in the operating room. But in cases of both topical phage use intraoperatively and for wounds, you have to be careful about having the phages diluted to too great a degree, because phages are more stable at higher concentrations. One of the challenges of the Phage study was that they were diluted in transit, and by the time they got to the patient, the actual titer of active phages was hundreds of times less than they anticipated because of loss of activity in that dilute transport solution.
Okay. So Adam, I'm not able to get Dr. Chipper, do you want to ask your question yourself? Because I think he has already covered the release of endotoxin and the effect on the patient. As per Dr. Schooley, it's not anything unique if you see in comparison to antibiotic. If antibiotic causes release of endotoxins, so do the phages. Do you want to ask this question so you can ask? Otherwise,
No, he has already answered it. Thank you so much. He's already answered it. Yeah.
Sure. And Adam Good also, if you could ask, because I'm not able to figure out what you're trying to say. If I may, do you want to speak your question, Adam Good? Or I'll just read it. Maybe Dr. Schooley, you can get this. I would love to be able to ask a question about how we create advanced hybrid active sites and mobile treatment facilities immediately as a human security necessity. Okay, maybe the phage libraries or phage banks with the medical facility are on a mobile facility so that it can reach the patients sooner.
Yeah, I think that we're increasingly seeing more and more robust phage banks being developed, and better and better libraries, IT libraries of understanding who has them. In our case, we collaborate very actively with a number of collaborators here in the US, each of whom works with one or two different bacterial pathogens and kind of specializes in phages. For example, Anthony Moresso's group is really very proficient in looking for phages, their activity against E. coli and Klebsiella. David Pride at UC San Diego is really quite good with enterococci. The Yale group is good at Pseudomonas, as is the Walter Reed group, and is the Pittsburgh group. So each group tends to develop their own area of expertise. They develop what one would call frequent flyers, which are phages that are turn out to be often active against clinical isolates, and those are generally available pretty quickly to screen. I'm hoping that as we develop more capability of being able to prepare clinical batches of phages and to store them, that some of these frequent flyers can be pre-prepared and stored and ready to go. Once they're made, they're easy to transport at 4 degrees, in other words, on wet ice from place to place, and FedEx does that very well. So you don't necessarily have to have them at every hospital in the country. You just have to have them in a place where FedEx can get to them and move them around. So I think that over the next several years, we're going to see more and more availability of libraries that can be quickly screened, and some of the more frequently used phages already prepared in batches that can be given post-haste to patients who need them.
Okay, so these are the last questions. Ubaidur Rahman wants to ask, when the phage treatment happens and bacteria lies and a lot of phages are released, so does it anyway negatively impact the patient? Because bulk of phages are released at the site, so do they have any effects? I mean, that's what
we don't have any evidence that it is a problem for patients. Phages, as I said, are things we live with, and when they get into the intravenous, into the vascular system, they're cleared by the reticuloendothelial system in the liver and spleen. And so when if they are released at a site of infection and enter the circulation, they'll get cleared out, chewed up, and reprocessed. The same thing happens when we give them intravenously, which is why we have to give high enough doses and frequently enough to be able to kind of get past the natural defense system in the liver and spleen.
Right. So now the general question from Gunaraj from Nepal. And one query we also have is that how do you confirm finally whether the phage is lytic or lysogenic? I mean, those of us who work on phages have this quandary all the time, because some are very clearly lytic, but some are like dubious. So genomically characterizing them is fine, but then you'll have to engineer it to make it lytic. But what is a short way of saying that this phage is lytic and hence good for therapy?
Again, that's something that we need to learn a lot more about than we do. I mean, we tend to make decisions on the basis of efficiency of plating and on plaque assays about whether it's really a clear plaque and that sort of thing. But what happens in vivo in the context of a wound with bacteria of different stages of growth and different pH and different immunological milieu, how is that important? We don't know, and we need to learn that, because we may be putting aside phages that may not look as crisp and clean and nasty in vitro that would do a much better job in vivo. And we're going to need to, again, as I said earlier, connect the dots between our laboratory observations and what happens in clinical settings.
So final question, I think it's on behalf of everyone, Gunaraj, where he wants to know what's the global, like we have been hearing about success stories, isolated cases, some countries have been doing for decades, but still there's a lot of skepticism. So his question is, how does the global landscape look regarding the acceptance and integration of phage therapy into mainstream medical practice?
Well, I think it's like everything else. Success breeds success. And, I can tell you that 7 years ago or 8 years ago now, when Stephanie asked if I'd consider this with Tom, I had to tell her I'm kind of a boring traditional infectious disease clinician. This is nuts, but we should try it because we're not getting anywhere with antibiotics, and off we went. So I started out extremely skeptically. I'm much less skeptical now, and I think that's true for many people in the infectious disease world, at least in the West. And I don't interact as much with the global community as many of you all do, but I've certainly sensed from the standpoint of clinicians much less skepticism as they've begun to read the literature and understand the biology. Pharma still, big pharma, is still very reluctant to jump into this. And I think part of it is because they're so conservative that they don't want to fail. And many of them are standing by the side doing what they often do, which is waiting to see if the biotechs can make some progress, and then they'll swoop in and buy the biotech. I wish pharma were more adventurous. They're not that adventurous with new antimicrobial discovery, which is one of the reasons that a lot of the mechanisms like Carbex and others have been put in place to try to incent more industrial engagement. But to be cynical, you can imagine, being a kind of a mid-level pharmaceutical executive or scientist going to your boss and saying, I've got this stuff that's been around for 100 years. It's failed consistently, but I want to put a couple $100 million into this. How about it? So that takes courage to do. And there aren't a lot of courageous people at that stage in their lives in big pharma these days. And I certainly understand where that's coming from, but I do think that if we are rational about how we evaluate these and use, as I said earlier, the same metrics we use to assess antibiotics, and some of these same people say, good grief, the cultures were negative just as rapidly. And in fact, the patient didn't relapse. Maybe that biofilm stuff means something. As people begin to look at some of the clinical responses and some of the microbiologic responses with endpoints they're used to seeing with antibiotics, I think it's going to help bring this back into the mainstream and away from the magic potion that has kind of plagued the field for so many years. That's certainly what I'm hoping we'll see over the next few years.
Okay, thank you so much. We really appreciate your time and answering all our questions so patiently and so eloquently. We were absolutely I think it was extremely, for those of us who are not us, I mean those who are joining, because we are doing this series particularly for from the clinician, so that we know that if you say, then there's a lot of weight, because it's not like something imaginative. You've yourself done the treatments and you've been doing, I think, a part of a clinical trial also. So what you say gives a lot of faith and a lot of encouragement to us who are trying to bring this therapy, if not mainstream, then at least to begin with, for compassionate grounds more often. So thank you so much. Really appreciate your time.
Thank you, and I hope you all have a good holiday season. Thanks very much. Bye-bye.